This is Unit 1 Macromolecules. To start off, we have carbohydrates. They are made of carbon, hydrogen, and oxygen always. Their sugars and polymers of sugar. They have carbonyl and hydroxyl groups. They are monosaccharides, or they're made of monosaccharides, simple sugars. These are units of CH2O, carbon and water, and then the most common monosaccharide is glucose. The glucose is nutrients and fuel for cells. And there's disaccharides, which are monosaccharides connected by covalent bonds, two monosaccharides. So this is half a disaccharide, two of them is one disaccharide. And then the most common disaccharide is sucrose, because sucrose is 2 glucose, as we know. Monomers of sucrose, like I just said, glucose, and also fructose. Plants transfer carbohydrates from roots to leaves in the form of sucrose. And now we're on to polysaccharides, which are storage there's storage polysaccharides, such as plants that store starch and animals that store glycogen. These are both used for storing glucose. Polysaccharides are made up of many monosaccharides, more than 2. And there are also structural polysaccharides, such as cellulose, which is a tough substance that's used for plant cell walls, and chitin, or is it chitin, I forget, which is the exoskeleton of arthropods. And now we're on to proteins. Proteins are made of carbon, hydrogen, oxygen, nitrogen, and sometimes sulfur. And they're basically the simplified version of a protein. It goes amino acid to peptides to polypeptides to a full protein. And proteins' shape always determines function. There are 20 different kind of amino groups. And each amino group, which is drawn here, has the R right here, which is a variable side chain. And each of them have a unique group, and it can be grouped as nonpolar, which is hydrophobic, hates water, not polar, hydrophilic, loves water, and charged/ionic, which is hydrophilic as well. Side chains interact with, the way the side chains interact determine the shape and function. Now over to this side, we have the primary and secondary structure. So the primary structure is determined by genes. The primary structure of a protein, of course, it dictates secondary and tertiary forms. This is just 2 and 3. And then there's the secondary structure. This coils and folds, as you can see. Not yet, but it will. It coils and folds to hydrolyze bonding, or dihydrose bonding, and it has B-planted sheets and like A helixes. And this is I skipped one. This is tertiary. This has 3D folding, and this is due to interactions of side chains of amino acids. Remember the side chains, how those react. Those cause a 3D folding. Remember, they determine shape and function. And now the interactions of bisulfide bridges reinforce this shape you see here. Now on to quaternary, which is the fourth protein structure. It is associated of 2 or more. It's an association of 2 or more polypeptides, and it is found only in some proteins. And now we're on to the functions of proteins. It's 5 major functions: antibody, enzyme, messenger, structural, and transport and storage. Antibody helps protect the body from foreign elements and disease. Enzymes, they carry out chemical reactions or assist in creating new molecules that catalyze the substrates. As in the other video, we talked about it. A messenger, it transmits signals, like hormones. We also talk about them. Structural, they provide structural support, such as we saw over there. And then there's transport and storage, and they bind to carry small atoms and molecules through the body. And that concludes the first 2 macromolecules.